So I'm going to call Professor Guilherme Oliveira to be part of the panel from CBE, Dr. Rayan and Dr. Newton Barbosa. And we'd like to thank the audience for joining us and our guests too. Rayan, Newton, Professor Guilherme, thank you for being here. Thank you, Gabi. Good morning. Good morning to everyone. Rayan, Guilherme. It's a pleasure once again to be here with you and to receive you, and the presentations were wonderful. I think that every day we discover that eDNA can work for more things, and we are incorporating these technologies on a daily basis. So I'm going to open up for you if you want to make a few comments. Yes, go ahead. Yeah, Rayan has his microphone closed, so I'm going to start. I'd like to thank you for the opportunity of being here. And here at ETV, we have used eDNA quite a bit for issues that were discussed here, but also in terms of biosafety. So we were able to look at environments, and you don't want to have people in caves, right? So we have this aspect as well that is important. And I have no doubt that it's a technology that adds a lot to the identification of invading species. And I think that the value that this event can bring, it can actually allow us to work together in the establishment of standards. That's one of the things I said. If everybody uses a different method, you know, it's not that one method is better than the other, but if we unify, it increases the acceptance of companies and the environmental organ in terms of our findings. But once again, thank you for the opportunity of being here with you. Good morning, everyone. Good morning, Guilherme. Thank you for the opportunity once again, talking about the work that we have done in this project. And this time we've had a few advances, and I agree with what Guilherme said. I think it's very important for us to have these moments. You know, as people wrote in the chat, I didn't know about it, I didn't understand it very well. So as we bring this, as we shed light on it, it becomes very important, and also in terms of application. And it goes beyond the invading species, and it could be differentiated tools, right, for us to be able to monitor the environment in general in a very rich manner and with a lot of information. Yes, and there's an article that came out last year, and we have this in the group. It's from Eva Lavas, and it's exactly what Guilherme said. One of the greatest challenges in the use of these technologies, and strongly in terms of public policies and Brazil and in Europe, it's really the standard, because that's the most difficult thing to do. And, you know, it demands constant conversation, and it's one of the objectives here at this forum. It's exactly to bring people together, bring several stakeholders together and researchers and companies, because we need to start talking about this in an equal manner. Even the databases, everybody needs the databases and libraries to be able to identify these things. So it's really very important. So I'm going to start. There's a few questions that came in, so I'm going to ask you the first questions. There are several that we have here today as well. So the first one has to do with what we just talked about. And so it's two questions from Veronica. And the first one is a question about, you know, if many companies think that the cost of investing in molecular biology is too high. And Guilherme, since you are inserted in a company. What is your opinion about this? Does it really make sense? Well, the issue of cost is really something to be considered. So as a research institute, we have to have infrastructure, and not only for eDNA, but for lots of things, that we use this technology. But it's another reason for us to talk about how we do these things in Brazil. And we basically spread out initiatives. Everybody has a little bit, but nobody has something bigger to be able to give a large step forward. And forums like this one help the community to group around an objective, a common objective. So we don't need to have 50 labs in Brazil to address the use of these technologies, or sometimes even in terms of sequencing, right? As in the case of the very important study that was made on mussels. So yeah, and even the service providers, it's important for them as well. So one of the ways of doing it is to have centralized databases. There are public databases, but the community, it's the different species and for the species that are interesting. So this can be an exercise. And I think that the approach for those who are going to finance this it's much stronger than the individual. So doing it individually. So it's really nice for this group. Yes, go ahead, Rayan. And thinking about what Guilherme said, I agree with this. I agree with this collaboration, because if we have a good reference center that provides these services, we buy this service and we can maintain this center feasible for the community. And I believe that it would reduce costs, because what we see a lot in terms of molecular biology, it is the high cost of molecular inputs, because lots of it is imported. We don't produce it here. So we really need to reduce these costs. And as soon as we have a good center for this service, we're able to improve this economic network. And the references, that's extremely important, because we work in this institute, and we have the analysis of eDNA, and we have one reference center, and it would really make it much easier for us. And when we talk about the genome, that was it's a subworld, a genetic subworld that we're in. So we really need to have these sequences available and making them easier to be accessed. So sometimes I see the databases and I get lost in the databases. It's not that simple. It's not so intuitive. So it's very important for us to be able to have this sort of perception. So not everybody is a molecular biologist, and sometimes people need the information, right? So that's exactly what we're talking about. Yes, and that makes it clear. In the article that I talked about, people talk a lot about the way in which the groups communicate. And sometimes you have opportunistic interactions, and that's what usually happens. The researchers meet here and there. We have these sustainable journeys like this one that can result in collaborative work. And that's basically what we should look for, right? Try and talk in the same language to be able to build the libraries. So that's really very important. So Guilherme, I saw you talking about the work in Carajás, and there's Pedrinho, who did a PhD with me, and when I was doing my PhD, and I was thinking, I came up with a question here. We could do these libraries through what we have in museums. Yeah, we have this because we have done this because, as you know better than me, it's not always easy to recover a plant. It's not as easy as we would like, right? And to do short reads, which are sequences from a library, it's possible to use what we have in museums. Of course, the efficiency is lower, but this is an approach we're using to verify some cases of plants that were collected once, so that's the advantage. Now, for animals, it's more complicated because it's not always stored in a correct manner. But for plants, it's quite possible. Yeah, plants get dry, right? And then using. And in the case of you being able to get information and identifying according to the eDNA, well, collected once. Well, in flora, no, because what we did in flora was a complete revisitation. So we really went through it. Yeah, it was an expedition. So it was more than well, it was more than 100 people involved. It was really something that is even difficult to do in other environments. So we didn't really have this case, but we've already had some curious cases of some plants that are rare. So we want to review some previous collections, and this is going to happen. Okay, that's very interesting. But in this case of the Itzicatas, there is a little bit of availability because they are stored inside environments, and there is a time to remove the DNA. Or even if they are stored for a long time, you can still do it. Well, it really varies according to the species. The fresh species, they're difficult to extract. There's everything in nature. So it varies, right? It depends on the conditions. And this is really something, it's on a case-by-case basis, right? We have to try it out and see if it works. That's really interesting. Now, I asked Veronica a question, and she has another one. And as you said in your presentation, Guilherme, you said that you have a patent of the system, and she wants to know what the greatest challenge is to the development of a product using molecular techniques and about technological maturity. So how can these techniques get to the market? Well, that's an interesting question. I don't think that the patent is really something that is going to provide a secret. So it has to be a service that is well done, because the sequence of DNA, this is public. So if you have a detection method and people maybe don't want to share it, but then I can go and do the sequencing myself. So it's not the DNA sequencing data that has the intellectual property. It's more the development of the detection technique. So you can do great work that works very well, and then I would have difficulty. So his service is going to be better than mine. So that's how you can get the market. And when it's eDNA, I think there is a strong line of computational analysis. We protect our products routinely, not because we think that there's no intention in terms of getting money. People can download it if they want. It's more in terms of for the companies. But once again, I think that you have to do the service for the market. So do the service in a robust manner. And there are some companies, and they are going to be successful if everything works out, because in terms of the support and environmental monitoring, technology is here to stay. So the companies are incorporating these technologies, and there is space for others. Can I just add something, Newton? Yes. Uh, I think that, first of all, we have to change our perspective in terms of research and company, this relationship that we have in Brazil. I think we still need encouragement of researchers. They have good ideas, but sometimes they don't have, encouragement. They don't have a way of encouraging them in the market. We don't have the financial resources either. And there are some who are starting to do this, and we've had good outcomes, and that's important. And taking advantage of this question, it's right. Please correct me if I'm wrong, Guilherme. So something that you said that called my attention, I have a lot of difficulty with information technology, right? Sometimes I understand the program, I know how it works. And then at a certain point, I just, block and I look for information. So, you know, I need a sequence, a specific sequence, and I couldn't find it. So I ask a collaborator, and in half an hour, he's able to do what I need because I don't have this, this knack for computers. And so it is easy to use, and it's relatively simple for those who don't work with information technology, and we can use it. Well, don't feel bad about not knowing. When we do multidisciplinary work, everybody knows about their area. So here in ETV, if it weren't for the team, we wouldn't be able to move along. But in the case of PIMBA, we're not a software house. We don't do computer programs, but PIMBA is a pipeline that is accessed. And to make the installation easier, we usually do everything in Dockers and containers. So you can download this virtual machine and install this machine so you don't have to install the whole environment, because sometimes it's more difficult than installing the program itself. So it comes with a very complete documentation about how to use, how it can be used. So I don't want to put it as a software that you find on the shelf. This is knowledge. And there's something else. This has to be done as part of the sale of a service. It's expected. Not everybody is going to have access and is going to be able to find sequences. The different sequences are in the public banks, right? So the boat system, for example, and accessing them is not necessarily trivial for those who don't work with this. So it's a different exercise that can be done with the community as well. So it's creating this reference database that is updated and just to make people's lives easier. I think this is really very relevant. And something that we do in PIMBA is to make it feasible. You don't have to download the whole thing. We have specific data banks. So for example, we have different layers, and this is for the plants and to identify this as closely as possible to the species. And then we go to the global bank, and this is, it's an African family, but, we're able to take a look. And we have this, we have these tricks. But anyway, going back to the reference sequence, if you're talking about an invading species, who is the reference there? The species, the native species, for example, the mussels would have to have a sample in Southeast Asia, or do I have to do sequences, for example, in Brazil, in Europe, in Asia? And what do you think about this? I think it's really complex, right? When we talk about invasions, I think they're all valid, because if you have an Asian one, it's better than not having anything. But the closer you are to the environment, for the environment in the region that you're analyzing, the better it will be, because you can have more specific markers for this, as in the case that you used. If you do a shotgun, it's a less of a problem. But I would create the references, the local references. Now, with vouchers placed on public collections, and certified by specialists and taxonomists. That's very important. I was thinking that maybe it's the case that we should start thinking about making this feasible, seeing if it's possible. But having a database like this one and managed by another state, a library, a public library available for everyone so that everybody can use it. I don't know if this is something possible. I think this idea is interesting, Newton, but it can be simplified, right? We don't have to think about a huge infrastructure or anything very complex. If you have a curator group says, okay, these are the species that we already have or we would like to have in Brazil. And then you can do automatic searches in the database to, it could be a file placed in FTP. And you don't need to have a structure. So the cost is very low and it's very relevant. And where should we actually look for these sequences? Yeah, it could be a hot site, right? From a university, for example. Yeah. Now, talking about this issue of development of the techniques, I have two questions for Rayan. And the first one is from Mariana, and she is asking about the challenges that you had in this standardization of land. Well, for that, I'd like to go back to this important point that you commented on, especially in the end of your presentation, Guilherme, the availability of materials and manuals. I think, Lamy, when we proposed this project, I thought that according to what comes in the articles, it seems to be very simple. So you get the DNA, you get a primer, and then, everything's going to be colored. And over the years, I've seen that that's not the case. It's not that easy. And when I see these articles, I really miss important details. I understand that today the journals want objectivity, reducing the number of words. But in the case of techniques, I think it's very important for us to have a detailed description. And it's very difficult to find these articles. Sometimes you find the base article with this technique, but since it is the basis and the technique evolved over time, the routine is not placed. And then when you go to the lab, you find these problems and there's no way to solve this problem. So in the case of LAMB, I really missed this manual, as Guilherme said, and it was thanks to the last panel that we had in the forum, Rubens and from Fiocruz, and this is definitely how we were able to move forward with the outcomes. We did several meetings and he talked to us about what could be done. So I really felt a lot of difficulty. I think that's the greatest problem we had before this collaboration. The articles are not enough to be able to carry out this technique directly in the lab. It's not that easy. So I think that this idea of creating manuals and creating these areas so that it will go to the interested public, because it's very important, because sometimes we really try and we try to get a good outcome, and sometimes it's because of very simple thing that changes the routine in the lab, and then with that you're able to do it. So I think that was the biggest challenge, not having a protocol that was really the reality. But I think today we're at a good point, and when we publish these outcomes, I'd like to detail this methodology, what we did in the lab, so that whoever wants to replicate the experiments will be able to reproduce this more easily. I think that's very important, and going back to the manuals that Guilherme talked about, and I think that's important. for bioinformatics and for DNA, eDNA even more, right? Because sometimes people don't have this expertise, but I want to do the work. So how do I start? And if you have this manual, I think it's a great opportunity. And sometimes we put this at a second level compared to the original article. And then, there's difficulty, right? And especially to go into the areas. I don't know if Guilherme wants to add something about this. Yeah, we felt need of a manual. And we use it a lot in the work of the consultants, right, that we hire for the collection of material to produce the different libraries. So we do the training, and we saw that it's not enough to do the training in the field. We have to create these standards. And something that is going to be necessary. It goes from the collection of the material until the informational inspection. So when you talk about the collection of some materials, people say, oh, so you have to take the icebox inside the cave, right? It's small details, but it makes all the difference. And the standardized procedures, they are extremely important. Okay, I'm going to ask a question about LAMP again, and it's from Clara. She talks about the possibility, if there is any light on this, to use multiplex LAMP. Are we able to have several colors appearing? Well, it's difficult, right? Well, I think that it's interesting. It's an interesting approach. And going back to what was discussed yesterday, one technique is not better than the other. It depends on our objective. So it's very important to be clear on this, what we want to use to get to the outcome we are expecting. So I think that is very interesting because you are saving time and freedom, and molecular biology makes a big difference. And the more you try, the less chance of contaminating the samples. And then you also gain time in terms of working in that environment. So it could be an advantage in terms of the lab. So the multiplex approach worries me because the lamp by itself uses 6 primers. One is huge. So imagine if these primers adhere and they can generate some kind of crossing that is negative. Of course, for me to treat this, I need to standardize that. It can be done in the lab, but for LAMP, it's going to be a little complex, and I would not use multiplex for a LAMP reaction. What I would do is place several sets of different primers, and I would do LAMP for these separately in the field. And also this issue about the color, right? Thinking about using immunofluorescence in the lab. And for example, one is red, one is green. When, I put them together, we get a yellow. So maybe we can get a mixed color to identify the presence of both. So it would be very interesting, but I'm really concerned about this crossed connection because I'm looking at 2 DNAs that I look at. And in the field, there's a lot. So I don't know if this could be a false positive or a false negative. It would increase the chances. I don't know if the LAMP multiplex would be interesting for this kind of diagnosis, right? When we're talking about the invading species in an environment. But it's an idea, and I think Clara is right, and we can investigate this, but we have to be very careful. And we also have to think that increasing these primers. For example, if I'm going to test 2 reactions, I would place 12 primers. And this can also diminish the sensitivity of my technique. And that's very important in the case of a low amount of DNA. So I think it is feasible as long as we standardize it very well, but I would not use this alternative. I would test both separately. I think the advantage would be bigger in the case of the lab, but it's a good question. Would you like to complement that, Guilherme? No, he was 100%. Great answer. Okay, I have a question from Renato, two questions. He is congratulating you on the presentations, and he was curious about what was used in the flora of Carajás. So he made the numbers go from 200 to 1,666 species. Yeah, I missed the audio here, but I think I understood the question. Would you like me to repeat? No. I think I can just let me know if I answer the wrong question. So the increase in the number of species, first of all, it's not new species necessarily. It's just species that in the traditional work had not been observed in the location. And this happens. I have a case where I found a flower on the ground, and it was a different sort of flower. And I collected the sample to do the DNA, and I took it over to our queen, Juliet, to be able to tell me what it is. And it's a plant that nobody had seen in Brazil in more than 80 years. So, we see this all the time. And if you don't check it out, you lose it. But, the yellow light is always on because the work that is done with taxonomists in very diverse environments, they suffer. It's not because people do a bad job. It's a difficult job without the references. It's really very complicated, right? For a taxonomist to identify a plant that doesn't have flowers. And, it's really very complicated. And in Carajás, right? Exactly. Yeah, there's a lot going on there. And lots of species that we collected. The taxonomic resolution was not well defined. And recently we described a tree that was in the park in everybody's face and nobody knew what it was. And this tree had been seen before in Guyana, and the taxonomists went to dig up literature, and they discovered some descriptions of this tree. And I'm calling your attention to another problem here. Doing this based on genetic data places all of the effort in the digital world, while doing this plant by plant and comparing, it's Herculean efforts. And for us, it makes a complete difference. And then you say, is it expensive? Yeah, but what is the cost of your time? What is the cost of not knowing about things? So there's this component. And also subject to errors, right? So when I worked over here in Serra do Cipó and Carajás, you have, the effort has to be much bigger. And the chances of not having a plant in the field is always much bigger, right?
Agora, quando a gente foi muito legal.
Now, it was really very nice when we had a team of taxonomists, a top team, which is very difficult to find, like Juliet. Yes, exactly. So this allowed us to make an exchange with them, because sometimes we came in with plants and they said, oh, so this plant, there's something different about it. And then we checked it out genetically. And sometimes we saw things genetically speaking. They say, okay, what you're talking about, and probably there's something else there. So it was really very interesting. And we knew, same thing, why? It was really very enriching. And the number of species, we have 1,106 species, but there are still some that we can add. It's very interesting. I also wanted to say that as from this genomic analysis for DNA, the moment that you recognize the plant that you haven't seen for so many years, when you do the opposite, then you go to the field afterwards, you can look at it differently. Oh, we saw this plant. Is it here? And you start observing and you do the opposite, right? It's directed to what you want to see in the field.
Mas eu acho que também ajuda
we have to do the analysis, of course, but I think it helps us to recognize this, and it can be very enriching depending on your objective with the sequence data in the lab. Yes, you're right. And there's an element that is good for discussion. It is very controversial in terms of that. The taxonomic operational units. So when we do eDNA, I don't know if you remember, but we find more than 560 OTUs in a camera, but it's 60 something. And what do we do with OTUs? In the traditional manner, you pretend they don't exist, but they're there. There's no way we can say they don't exist. So I don't know, looking at the world of genomics, and this is in a very fluid manner. When I know about taxonomy, people, they want names, and changing names is very complicated. But in genomics, we have the tax ID, and it's one today, different one tomorrow. That's fine. And my opinion is that it should be incorporated. Of course, at the end of the day, I want the taxonomist to give me a name to that plant, for that plant, right? And the specification as well. But while I'm not there, I can use this data, right? I don't have to throw it away. And when you do monitoring and you see that you are floating around the identification, and then suddenly you see something different, that really calls your attention, even if you don't know the name of the plant. So something happened in that environment. And I think this layer of digital analysis, and also the feasibility of reanalyzing, because maybe today I don't have all the references, but in 5 years' time, I can actually improve my database. I can do that. Yeah. Actually, we've seen several articles, and I talked about this yesterday. The production, it's because it's changing. It's not that the plant is becoming extinct. It's changing, and many times the taxonomists have difficulty with this new paradigm. And I noticed that taxonomy and genomics, they're moving around together, and sometimes people are not able to get into this rhythm. But we have to use the dynamics that genomics brings. What you said, right? It's much faster, and there are less mistakes compared to traditional taxonomy. Well, a taxonomy is more necessary than ever, right? More than what we can write. But just because of the preciousness of these people's brains, we have to direct the work of these professionals to the areas that are really complicated, and not just try and re-identify plants and waste time with that. And I can do that without thinking. So I think this values the work of taxonomists, and I'm not going to replace him, of course. Yes, of course, that's it. Since you talk about fish, you talked about fish, and Renato asked a question about fish. So he said the following about the DNA of fish. You showed that the 3 made up with 16S, there are divergences in terms of the morphological analysis. So maybe we could use a quick evolution marker to try and check out the different groups that are taxonomically similar. Yes, you're absolutely right. And this is our open door. When we detect something that doesn't really feel right, we routinely use EPX2. And in the case of plants, if we get literature, European and American literature, and we applied it in the Amazon, it wouldn't work out. We need markers for the Amazon. It's not the same markers that we have, and it's important. It's not the same importance, and it's not the same relevance as we have in Europe. So that's the entry door. We identify the possibility of inconsistency, and then we go deeper into it. So, for example, we already have the mitogenoma for these species, and the way we do this using the sequencing, we make analysis that are based on the frequency of the genomic data produced. And this gives us great resolution, genomic resolution associated to morphology. And what we did recently to try and place morphology in the digital world, we acquired all the species where we have some kind of question. We do the CT scan of the species, and now the next step is to apply artificial intelligence to classify the images. And this is also when the species is going to end up in the collection. It's going to be well kept, well stored, but we have all of the digital information available. So it definitely accelerates what we do to analyze this. So you're absolutely right in what you're saying. Would you like to add something, Rayan? No? No, because when you talk about 16S, I only think about the work that we did on metagenoma that Mara's going to discuss tomorrow, sorry. And that's as far as I go. So let me ask you a question now. It's from Roberta Caldeira. So she's thanking you for the presentations. And she's talking about the use of LAMP PCR in the field. I'd like to know what kind of stain is going to be used. So CBER or CYBER, yeah, it's called CYBER. It is a DNA stain, and there's a problem because we have other organisms in high quantity, right? So first of all, my answer is I don't know, Roberta, because we don't know this yet, because whenever we proposed LAMP as a research project, my greatest despair in my naive head, I thought, okay, we go to the lab, then we go to the field, and that's it. And I was always very desperate about that. How am I going to make this happen in the field? And how am I going to make it feasible in the field? But then I found these barriers, and I'm still in the lab, right? But we have discussed this a lot, have discussed cyber a lot. It is fluorescent, and there is an ultraviolet intensity to change the color. So in fact, I don't think that cyber is the best stain for the field, the way that you put it there. But thinking about the lab where we receive a lot of water samples, and sometimes you have one larva in X liters of water, so we can use this technique. And also, there are many experiments that were done with cyber. So that's the easiest way that we found to standardize. For the field, we are still studying what is going to be the best mechanism, if it is going to be the color, the way that we see it. And we have talked about using some sites based on magnesium. They change the color, and the change in pH has also been very interesting. It's an alternative. So we're still discussing what the best method will be, because, what we have in the field is deal with this large quantity of DNA in the water, and how can we make this extraction in the field in an environment that doesn't really have a lab support, and how to reveal this with these issues about multiple DNAs in the water. Although we are standardizing this in the lab, we want to do the experiment of collecting it in the field and testing it in the water that comes in from the DNA in the mussels, and there are other DNAs to check how the behavior of LAMP PCR is going to be. That's the next step we're going to follow. So unfortunately, I don't really have an answer for you about the stain, but we are studying several possibilities, but it's a very good question. Yeah, I think you also answered Julia's question. She's asking about how the standardization of LAMP is working out. Do you think we're close to real-time monitoring? I think, well, I sort of answered, and I had some answers in my presentation about sensitivity, the reaction time. I was able to do some experiments like that. So we saw that in 60 minutes, the sensitivity is the same, 0.01. and nanograms of DNA in the solution. But to say that, well, we are close in the field, but if it's going to work in the field, that's something we still have to check out. And we would like to have Dr. Evangelhos helping us, and he's leading this part of the DNA extraction in the field, the feasibility in the field, together with the knowledge that we have from Fiocruz to bring this and, get what we have together with this and make it into a field protocol. But I think we're moving forward. That's important. Great. There's a very interesting question for Guilherme from Daniel. Let me read it. So up to now, in terms of eDNA, it became very clear that this tool is used for the detection of species that are present or not. And we also have biometrics of individuals and sampling techniques that give us information about the diet, the reproductive status, the temporal and spatial info, and the relationships of these variations with physical and chemical factors in the system. Some of the information, could that be extracted from the environmental DNA, or is it specific only to detect the presence or absence of the species? That's a great question. I'm also curious about it. Well, using a single marker, for example, you have limits in terms of the number of variables in the sequence that you're going to use. We have, for example, the case of a species of macrophage. It has 2 SNPs that are diagnostic for it in the different markers that we use. So then you have a very low resolution to say something about the genetic diversity of the plant and things like that. However, there are 2 things that are happening. The first one is that we can test it, and instead of using long sequences, because we have a sequencer, TechBio, that allows us to make these long sequences and with a lot of equality. So then you begin to go into an area where you're able to ask these additional questions because you have variations and they allow us to say something about genetic variation in this population and so on. Of course, nothing compared to doing a complete genomic assessment. Now, with regard to the diet, if you consider if you collect, fecal samples, as we've done for iguanas and birds, you can say something about what these animals are eating. So we're going to start, for example, one for bees to check the flowers that are visited. So you have different ways of doing this. And we also have what is collected in the air. So it's a lot of interesting things that we can do. It does not solve all of our problems. There are limitations in the technology, even when it's done very well. There are things that we're not able to obtain information only from that. But the ecological information, there's a lot we can do, especially when you accumulate the data and there's a perspective which is not individualized, right, from one trial. I think this is greater motivation for us to use methods, standardized methods. So imagine how great it would be to have the biota, the aquatic biota of Brazil, and everybody's using the same method and with a unified visualization. That's a dream, right? You can get a lot of information, but we do have limitations. Now, the temporal and spatial dissemination, if you see an OTU coming up and you do regular monitoring, it can disappear and then there's an alert. And if you do this in several places as well, yes. I can spend another half hour only talking about this. Yeah, Alice Valentini yesterday said that she used mail to know which plants were visited. She used honey, right, to to check what plants were visited by the bees. So we can put the bees there, right? If you put the bee there, you will know, at least most of the flowers. There are many things that can come up, right? There are still many possibilities when we study. Yeah, the DNA collectors, the bees, predators, and yeah, exactly. But I also think about conservation because, if this bee, for example, there are 10 species and the bee goes to 7. So, this bee is going to die if it doesn't have all 7, and the other 3 it never goes to. So you're able to notice the dynamics of the ecosystem from this DNA collection. So there's a lot of information about conservation of the environment. Or out of 10, the bee goes to 1. So it's a very specific relationship. So if you kill the plant or the bee, you're going to, change the ecosystem. So conservation, is definitely sustainability, and it becomes incredible thinking about that. It's a very important tool that we have. And then I'm thinking exactly about what Guilherme said, the floods, right? And then a fish gets to a lake that they've never been in before, the hydrodynamics of the channels. And it's something that we always talk about, and it's very difficult to investigate. So, for example, you have new microorganisms in this fish, and it could be a pathogen for some species of fish. So everybody knows this, and the dynamics change, and that's very important to know. So I think it's a very good tool for conservation and also in terms of ecological relationships. Yes, so we have Afonso from Uberaba and Paulo, and opening up several regions with parasites. And it's a great effort that could be done with eDNA. You find cercaria as well, the DNA in the DNA of animals. So morphologically, we can identify some cercaria, but others we can't. And also, when we do the barcode per species, then you have the information about the animals that live there. So we see a lot of parasites, we see mites, and so on. Yeah, and exactly. Ticks and, just like mussels, as if it were the Troy Channel that opened up several species that we haven't looked at. And these species are maybe invading as well, right? And this will affect the environment, even more than what we see there. And how are they changing the dynamics of the population? So sometimes we say, oh, so we have other species and then mussels came in, but maybe it's not the mussels, maybe it's a parasite that it brought, and we haven't looked at it yet. So I think it's very important for us to start thinking about this sort of approach for conservation, management, and interaction of the species. So it's important to look at this field. Since we're talking about conservation, we have a question from Fabiano. He is congratulating the speakers, both speakers, and he says the following: the rupture of dams in mines, in the mining areas of Minas Gerais, talks about good previous samples. When we talk about restoration and even for the dimensioning of the judicial areas. Do you think that this technology can improve the current diagnosis? Are we ready to place this technique into practice in these studies, commonly called baseline diagnostic methods? Fabian, that's what I dream of, for us to have a baseline for all areas. And over here, I can say, according to the work that was done in Carajás, it is the search for this robust baseline that we're going for. So if you have the perspective of an entrepreneur, so you plan something, so it costs billions in Carajás, and then after 3 years of development, you discover that there is a plant or a fish that was not identified, and this can cause serious problems. So out of this robust baseline, I think it's everybody's dream. Whoever wants to generate operational safety for the business and everybody who is involved with conservation of the species. So that's it. We have done this not only in terms, in biological terms, but also chemically speaking, because the chemical baseline is important for you to know if there is some change because of something that happened. When we see in the environments, there are geochemical variations in places. So definitely you got it right. Rayan, would you like to add anything to that? No, I'm just thinking about this question. It's a very important question. And, you know, going back to what we talked about in the beginning, we have the history of the environment and how much information did we lose before the rupture of those dams, right? What was the environment like before? So I think that this information, this, genetic care in the environment, there's still a lot to do in Brazil. So we're bringing it to the current moment. So we test very little COVID. We also test our environment very little. We don't know what is there. And when this information is lost because of a modification in the environment, some limitation, so we don't have this history. And when we make this kind of movement, knowing what is in the environment, it's exactly what Guilherme said. So it is eternalized in the database. It is available and it's easier to revisit and to know what is coming in, what is leaving, and basically following up the dynamics. I think it's important for us to have this historical relationship, not only with the human lines, but also with genetics. And we don't really invest in this sort of movement with regard to the environment, also because it's a new technology, right? So I think it's important for us to maintain or to have this in mind, a lot of work that is supposed to happen, and looking at the populational and environmental dynamics. Yeah, in fact, the diagnosis is going to be a lot more complex. We have the growth of species that we have in Carajás. It went from 200 to more than 1,000, right? Yeah, that's right. So our time is coming to an end. I'm going to ask you a last question, since we didn't talk about this. It's a question from Renato to Rayan. So he's congratulating you on your presentation. And the question is the following. I would like to know if looking at the taxonomic shadow that is there, do you think it would be a good alternative to answer the questions and to provide sufficient information to be able to understand the relationships between the populations? How do you think we could apply this technique for an organism that is not really seen in Brazil, not really sampled in Brazil? Well, that's a very good question, but it's very complex. I think that compared to mussels, the studies have advanced quite a bit. We have the genome, and we also have some studies with proteins, very advanced morphology with the mussels. And if you look in literature, you have almost nothing. It's not only in Brazil, it's around the world. And as Lucy, our collaborator, usually say, it's gray literature. We don't really know this organism around the world. So I think it would be interesting to do the phylogenoma, because it would help us to try and solve the issue of cryptic species. So, are there more people and so on? And that would help. And we can also look for 3 invaders and what favors this invasion, because the salt in the environment is still very doubtful. We see this species coming up in saline environments, but they adapt very easily. So maybe it is naturally a species that can adapt the amount of salt. We see that temperature influences it more in terms of its growth rate. So I think that in genomics, we can find phylogenetic answers as well as these adaptations in the environment. So I think that's very relevant. And I think that for Brazil, the main point is, first of all, the identification. So knowing where this organism is, because it is identified as a plant because it really looks like a macrophyte. It looks like an algae, and sometimes it is mixed up with the mussels. So that's very complex. And they say, okay, that's an animal. So the first thing is to know how to recognize and knowing where the populations are in Brazil. And we have to make this effort to try and understand this genetic aspect in Brazil. And then I would like to make an appeal to those who are watching us. Please contact us. You can send samples because we really need to know where these Brazilian populations are for us to be able to make plans to understand this animal and to control it that is still under notified in the country. And we definitely understand very little of the ecology, the biology, and the genetics. So if you know about it, please send it over. You can contact us and make this feasible. We are very interested. Thank you. There's a last question here. I have to ask Guilherme this question because I like this topic. It's about citizen science. What is the level of training that somebody needs to be able to collect these samples with LAMP and DNA? So what about the initiatives from other countries? So citizen science. What is the possibility of this happening here for the analysis in Brazil? That's a very good question. Yes, very nice question. The collection is not difficult. I can do it myself. For plants, we normally use a solution with salt that we can make at home, and we use a cool chain for animals. It's not complicated, but it's also something you can do. You can have places for collection. It would be incredible to have citizens science because the capacity of samples that we have is unlimited, right? So there are so many newts around and so many newtons around to be able to collect plants, right? And if you look at the population, it's 25 million inhabitants. Imagine a fraction of these people collecting materials from places I don't even dream about. That would be great. And something that we're doing in this sense, because we are structuring a plan and using Carajás as a test in the field to take these portable sequencers to the schools and to teach the children to make the collections. And technology is still being developed, but I think it is going to become good, and we can decentralize that and democratize, and then we can show Brazil, because there's a lot to be shown here. That's true. That's true. Just to add on to that, I think that once again, I think it's important to have a manual for that. If you have a well-built manual that is available, and if I follow that manual, I'm going to be able to make the collection to be able to guarantee the integrity of the DNA. So I think that we're back to asking researchers to make their manuals, to make them available, and it's important. And from this meeting, we're able to reach society, as Guilherme said, and I think that's very important for us to get support for this kind of work being developed. Yeah, people don't support what they don't understand. And as soon as we bring this over to the community, to society, we're able to get greater support as a whole. That's right. So just like everything in life, there's an end to everything in life. I hope it's also the beginning of something more sustainable and collaborative work within this topic. And we really have to do this sort of thing. So thank you, Guilherme. Thank you, Rayan, for your presentations and for dedicating your time to talk to us today. I know that time is difficult. I know it's difficult. Thank you very much. for today. I think it was great. Thank you very much. Thank you. And it's rare to have time to do this. It's great. I would also like to thank you on behalf of CBE that I'm also part of. Guilherme, it was great. It was a great presentation. So I think it was a wonderful morning, a lot of learning. Thank you. Thank you for your time and thank you for your availability and for discussing this topic with us. Thank you very much. Thank you, guys. So that was excellent. And I'm going to repeat this. I have to thank you. Thank you, Rayan. Thank you, Newton. And special thanks to Professor Guilherme. These events are very good and we learn about researchers. And in this panel today, we talked about collaboration, which is so important and we have tried to do it. So CBE has been trying to do this for a while to get collaboration and even with the participation of some of the audience here. We're going to continue these initiatives, collaboration initiatives. So hopefully this won't be our last meeting. The next one will be even better. Thank you, Guilherme, for your presence. And we'll be back tomorrow, Wednesday at 10 a.m. with more presentations and another roundtable. And tomorrow we're going to have Dr. Mariana de Paula Reis from CBEIH, Dr. Verhoeven Vosjespi, and Dr. Natrik Swan. They are both from Nature Metrics in the UK. And I would like to thank everybody for joining, the audience as well, and hope to see you soon tomorrow. And hugs to everyone. Have a great day.